I'm Barbara Sorkin from Office of Dietary Supplements, and it's my great pleasure this morning to introduce the first speaker first. My Office of Dietary Supplements colleague, Dr. Cindy Davis. Cindy received her Bachelor of Science from Cornell University and her Ph.D. in Nutrition from the University of Wisconsin-Madison with a minor in human cancer biology. She did postdoctoral training at NCI and then returned later to NCI and came to the Office of Dietary Supplements from there. She directs our grants and extramural activities at the Office of Dietary Supplements.
Well, great. Thank you very much for that very kind introduction. It's my pleasure to be here today. What I'd like to do is talk a little bit about the relationship between diet, the microbiome, and health. So what I'd like to do is give a little bit of background on what is the microbiome, what is the evidence that diet can influence the microbiome, and conversely, how can the microbiome influence the response to dietary components? And interwoven throughout this is what is the relationship between diet, the microbiome, and disease risk. So humans actually have been considered superorganisms, and that's because we're a composite of many different species. In addition to our human or mammalian or eukaryotic cells, there are also bacterial, viral, archaeal cells, fungi, phage, and in fact, there are estimates that there are anywhere between equal numbers to up to 10 times more microbial cells than humans. The most recent estimates suggest that there are 1.3 bacterial cells for every human cells, but that isn't considering the phage, the fungi, and other microbes. When we talk about the microbiota, we're talking about the microbes in a specific niche. So the gut microbiota are the microbes in our gastrointestinal tract. And in fact, that's about 100 trillion organisms. When we talk about the microbiome, we're talking about their collective genome. And in fact, there are about 100 times as many genes from microbes as there are human genes. You might also hear the term metagenome, and that's a combination of both the microbial and the human genes, or metagenomics, and that's the study of both the microbial and human genes together. So what do microbes do for us? Well, they have many important functions. They provide the ability to harvest nutrients. They produce additional energy otherwise inaccessible to the host, and that's mainly through the fermentation of soluble fiber to the short-chain fatty acids, acetate, butyrate, and propionate. They produce vitamins such as folate, biotin, and vitamin K. They metabolize carcinogens, such as the heterocyclic amines formed in food during cooking. They prevent colonizations by pathogens, and they assist in the development of a mature immune system. In the limit of time, I'm only going to talk about the relationship between the microbes and diet. So what is the evidence to suggest that diet can influence microbial profiles? Well, the first evidence came from looking at the microbial composition in globally distinct populations, and I'll show the landmark study on the next slide. But there's also evidence to suggest that long-term food pattern consumption can influence the microbial composition, and this has led to the term enterotypes, and it's the idea that people that eat similar diets have similar microbial composition regardless of where they live, their age, their gender. And the two main enterotypes are the Bacteroides enterotype, which is in people that eat a lot of protein and fat, and the Prevotella genotype, which is in people that eat a lot of dietary fiber. The other source of evidence is from short-term intervention studies, including low versus high fiber diets, animal versus plant food sources, and macronutrient ratios. So this is a landmark study that was first published suggesting that there's a relationship between what people consume and their microbial composition. In this study, they compared the microbial composition in children from Africa with those in Italy. As you can see in Africa, the main microbes are Bacteroides, particularly Prevotella and Xylanobacter. In contrast, in Europe, the predominants are Firmicutes. Why this is actually important is the two main Bacteroides, Prevotella and Xylanobacter, actually have specific enzymes present in them that allow them to hydrolyze cellulose and xylan, which allows them to actually extract as much energy as possible from the very high fiber diet that these children consume. So since they have a very low intake of energy, it can maximize the amount of energy that they can get from their diet. There's also evidence from short-term feeding study. In this study, 10 humans in a crossover design were fed either a plant-based diet or an animal-based diet. Now, I realize this is a very complicated slide, so I'll quickly walk you through the highlights. The plant-based diet is shown in green, the animal-based diet is shown in red, and their feces were collected for 4 days and analyzed. This is probably the most important part of the figure, where they look at the similarity and differences in microbial composition among the different individuals among the different days. And what I'd like to highlight is you notice that all the red tend to clump together and that all the green tend to clump together, suggesting that diet is actually more important and that when they're on these extreme diets, that their microbial composition changes and that it's more similar. So as I said, the short-term feeding alters microbial community structure and it overwhelms inter-individual differences in gene expression. And in fact, it modifies specific metabolic pathways. This idea that diet dominates host genotype in shaping the gut microbiota has also been shown in a number of animal studies. I don't usually go from the human studies to the animal studies, but the human study was actually published first. And in this study, they compared 5 inbred and more than 200 outbred mouse strains that were fed either a high-fat, high-sugar diet or low-fat, high-fiber diet, and they looked at the microbial composition. The results are shown here in this principal component analysis that looks at similarities between different samples. You can see that all the mice, when they were fed the high-fat, high-sugar diet, clumped together, and all those, when they were fed the low-fat, high-fiber, clumped together. And this was specifically due to an increase in Bacteroidetes when they were on the low-fat, high-fiber diet, and an increased Firmicutes when they were on the high-sugar, high-fat diet. So how can we use this information? What type of dietary modifications can we do to influence the microbial composition? Well, the first is the use of probiotics, and these are foods or dietary supplements that contain live bacteria, usually lactobacillus or bifidobacteria. Prebiotics, which are non-digestible food ingredients, which can selectively stimulate the growth of gut bacteria. Examples include inulin, other oligosaccharides, lactulose, resistant starch, and in fact, a number of dietary fibers have also been shown to exert a prebiotic effect. A symbiotic is a combination of a probiotic with a prebiotic, and the idea is that the prebiotic would actually be a food source or an energy source for the probiotic. And in fact, other dietary factors can also influence the microbial composition. Microbes are actually able to metabolize a lot of the polyphenols present in tea, cocoa, and wine and use those as an energy source. And many spices are actually antimicrobial, and that's why they've been used for centuries in food preservation, because they actually inhibit the growth of many different microbes. So I mentioned probiotics. The World Health Organization defines probiotics as live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. The key parts in the definition of probiotics are live, adequate amounts, and health benefit. These include everything from food to dietary supplements to prescription drugs. And examples of typical probiotics include lactobacillus and bifidobacterium. But actually, yeast is often sold as a probiotic. And other bacteria, such as E. coli and bacillus, we often think of E. coli as being pathogenic, but it actually depends on the specific strain, and there are large differences, and there are actually some that are being sold as probiotics. Fermented food and beverages typically do not contain live cultures due to the heat treatment, filtration, or food processing to improve shelf life. Hence, they are not true probiotics. Examples include sourdough bread, fermented meat, sauerkraut, wine and beer, and vinegar. Fermented dairy products include yogurts, natural cheese, buttermilk, kefir. And again, these may or may not be considered probiotic. And whether they are would depend on the bacterial levels when eaten and whether the bacteria have been shown to confer health benefits. Another important source of probiotics is dietary supplements. Many dietary supplement probiotics contain more than 1 billion live cells per gram, and in fact, many contain upwards of 10 billion live cells per gram. And they're probably better in terms of their efficaciousness if they have multiple strains of bacteria. This is data from the Nutrition Business Journal, which looks at the sales of dietary supplements in the United States. And this is data published in 2017, which looks at sales in 2016. That year, probiotics were the third most popular dietary supplement. Sales were greater than $1.8 billion per year, and it increased over 17% between 2015 and 2016. In fact, there are many proposed health benefits of probiotics because they potentially can normalize the intestinal microbiota composition. This can lead to colonization resistance and suppression of both endogenous and exogenous pathogens. It can control irritable bowel syndrome, inflammatory bowel diseases, and allergy symptoms. And they also have metabolic effects, which can affect colon cancer risk, lactose tolerance, and serum cholesterol levels. So what is the evidence to really suggest that probiotics do have these health effects? I'm going to utilize one meta-analysis to kind of show what are the strengths and weaknesses of some of the science out there related to probiotics. And I chose this one because it was published only about 2 months ago. In this study, they looked at studies that looked at the relationship between probiotics and BMI. They identified 13 subjects. And when they summarized the data, they found that individuals that consumed probiotics compared to those that didn't had a 0.27 decrease in BMI. And this was statistically significant. So of course the headline is, probiotics are beneficial for your weight. I want to point out though, that there's inconsistency in the studies. As you can see, some are on the left side, and others are statistically significant on the right side. And I also want to put it in perspective. Most of the studies are of short duration. They're small sample size and are actually underpowered to actually look at the relationship. Even more important, they utilize different probiotics. Probiotics vary depending on the bacteria that's being studied, or if it even is a bacteria. And in fact, they also vary in terms of the matrix they use, the doses they use. We can go on and on. Most of the trials are not pre-registered, but perhaps the most important point is that this effect is not clinically significant. I said that there was a change in BMI of 0.27. However, if they actually limit the studies to only those individuals that were either obese or overweight to begin with, they found that the change was halved. So there was only a change in BMI of 0.14. I don't think anyone would think that that's a really important change. So where is the science related to probiotics? There is preliminary evidence that some probiotics are helpful preventing diarrhea caused by infections and antibiotics and improving symptoms of irritable bowel disease. However, the US FDA has not approved any probiotics for preventing or treating any health problem. Probiotic supplements should contain at least 1 billion live cells per gram and are probably better if there are multiple strains of bacteria. If people are generally healthy, probiotics do have a good safety record. However, there have been reports linking probiotics to severe health effects, such as dangerous infections in people with weakened immune systems. What we don't know is which probiotics are helpful and which are not. Not all probiotics have the same effect, and the effects are likely strain-specific. How much of the probiotic people should take? And who would most likely benefit from taking probiotics? To be honest, whether a probiotic is beneficial for a specific individual probably depends on the background microbiota composition. However, this is something that's usually not taken into effect. As I said, there's also evidence to suggest that many different dietary polyphenols can influence the microbiota consumption. This was some studies that I was involved in where we looked at the bacterial abundance after consumption of cocoflavanols. And we found that in pigs given the highest dose of cocoflavanols, there was a significant increase in the amount of bifidobacterium in their feces and lactobacillus in their colon. Well, it's easy to say your microbes changed, but the question is, does it have a biological effect? And we actually saw that cocoa powder consumption decreased the expression of TNF-alpha and toll-like receptors 2, 4, and 9 gene expression, showing that it was actually having an anti-inflammatory effect. Well, I've mentioned how various dietary components can influence the number and types of microbes. However, this is a two-sided relationship, and I'd like to switch gears now and talk about the relationship of how the specific microbes that are present can influence the response to dietary components. Bacteria can produce new compounds from a number of food components. A few of the dietary components that are metabolized by gut microbes and their metabolites are shown here. In the limit of time, I'll only focus on the top two, dietary fiber, choline, and carnitine. But I should say, oftentimes, these microbial metabolites are more biologically active than the parent compound. So one of the dietary components that has been linked with health effects is dietary fiber. Dietary fiber has been associated with the decreased risk of colon cancer. When the American Institute of Cancer Research and the World Cancer Research Fund summarized the epidemiologic evidence related to dietary fiber and cancer, they found that for every 10-gram intake in dietary fiber per day, there was a 10% decrease in the risk of developing colon cancer. Moreover, this effect was linear. Dietary fibers are fermented by colonic bacteria to form short-chain fatty acids, including butyrate, acetate, and propionate. Butyrate is the most widely studied and the preferred energy source of colonocytes. And in fact, it has differential effects in normal versus cancer cells, which I'll show on the next slide. So this cartoon shows the relationship between dietary fiber and colon cancer. Dietary fiber can be divided into both soluble and insoluble dietary fiber. Insoluble fiber actually increases the colonic transit time, so there's less times for carcinogens to be in contact with the colonic cells. In contrast, soluble fiber is fermented to short-chain fatty acid, the most important of which is butyrate. In normal cells, butyrate serves as an energy source, so the colonocytes utilize it and actually increase proliferation because of its effects on energy, and it also increases differentiation. In contrast, in cancer cells, they undergo something called the Warburg effect. And what this means is they actually start utilizing glucose as their preferred energy source. So since they're already utilizing the glucose for energy, it means that the butyrate can affect other molecular targets within the cell. And the most important molecular target is HDAC, or histone deacetylase activity. And what it actually does is inhibits HDAC, which leads to decreased proliferation and increased apoptosis. There's also evidence to suggest that dietary fiber can affect bacterial diversity. It was found that when mice were fed a high-fat diet, a high-fiber diet, there's a large amount of bacterial diversity. If they're maintained on this high-fiber diet for multiple generations, they maintain that bacterial diversity. In contrast, if they're switched to a low-fiber diet, there's a decrease in microbial diversity, and over generations, there's a further and further reduction in diversity. If they're switched to a high-fiber diet from the low-fiber diet, there is some recovery, but not complete recovery, suggesting that you're actually having extinction of certain microbial species. Well, why is this important? Well, we know that intake of dietary fiber has been associated with decreased risk of cancer, inflammatory bowel disease, diabetes, but we always don't know the mechanisms. One of the things we do know that is in all of these diseases, there is actually a decrease in microbial diversity. So I would postulate that one of the mechanisms where dietary fiber is working to decrease the risk of many of these different diseases is through its effects on promoting microbial diversity. There's also evidence to suggest that dietary fiber can protect the mucus barrier. In this study, gnotobiotic mice, that is, mice that actually lack microbes, were colonized with a cocktail of human microbes. This was a cocktail of 14 different microbes that represent the 5 major phylum in humans. And then they were either put on a fiber supplemented or a fiber deprived diet and infected with an enteric pathogen. And what they found was that mice fed the high fiber diet have more fiber degrading bacteria, a thick mucus lining, and are actually protected from the colonization with Citrobacter rodentium. In contrast, animals that were fed the fiber deprived diet actually promoted expansion and activity of colonic mucosa degrading bacteria, particularly Akkermansia, and they are more susceptible to the rhododendron infection and actually develop colitis. Why this study is so important is all the mice were colonized with the exact same bacteria. And what this means is that the availability of potential food sources actually influences the growth of different bacteria and can affect the disease susceptibility. So even if people have the exact same microbes, what they eat can influence whether they might be susceptible to various diseases. There's also evidence to suggest that there's a relationship between diet, microbial metabolism, and cardiovascular disease. Dietary carnitine, which is present in meat, and phosphatidylcholine and choline, which are present in cheese and eggs, can be metabolized by gut bacteria to form trimethylamine. The trimethylamine travels to the liver where it's metabolized to trimethylamine oxide. And in both human and animal studies, this formation of TMAO has been associated with increased risk of a number of different diseases, including heart attack, stroke, cancer, obesity, etc. But you know, it's so easy to look at single nutrients in composition, and we really need to be looking at the totality of the diet. So this study looked at the relationship between dietary allicin and carnitine in the production of trimethylamine oxide. Mice were put on either a chow diet, a diet supplemented with carnitine, carnitine and allicin, or the chow diet and allicin. And for those of you that don't know, allicin is one of the bioactive sulfur compounds present in garlic. The animals were on the diet for 6 weeks, and then they were given a carnitine challenge. And the results are shown here, and I'll just take you to the top slide. If you look at the red diet, the animals that were fed carnitine, there's a large increase in the production of TMAO after the challenge. In contrast, if you look at the yellow group, which is the group that got carnitine and allicin, the allicin actually inhibited the increase in TMAO production after the carnitine exposure, suggesting that we really need to look at the totality of the diet. And I'll give you another example on this. I think most people have heard about the French paradox, the whole idea that people in France drink a lot of red wine, eat a lot of meat, but have a decreased risk of cardiovascular disease. Well, there's also evidence to suggest from animal studies that resveratrol has a similar effect and actually inhibits the production of TMAO. So perhaps this inhibition of TMAO production by resveratrol is actually contributing to the French paradox. Another important health effect is obesity. As we all know, obesity has been increasing over the years in the United States and is also associated or has been suggested to be mediated by changes in the microbiome. You know, it's something that's happening more and more to all of us. And calorie intake is one of those things that more is not necessarily better. But interestingly, as we all know, some animals can eat more and grow more than others. But there are actually some animals that can eat more and grow less. And this is germ-free animals. Those are the animals that lack a microbiome. Early studies showed that when you compare germ-free animals to conventional animals, The germ-free animals had 42% less body fat, despite the fact that they consumed 29% more chow than conventional animals. However, mice practice something called coprophagy. That means that they'll actually eat each other's feces. And if you take these germ-free animals and put them in the cage with the conventional animals, within 2 weeks, their body fat and their chow consumption are similar to the conventional animals, suggesting that these effects are being mediated through the microbiome. There's also evidence from transplantation studies to suggest that the microbiome in obese animals is different than those in lean animals, and it can be transferred. In these 3 different studies, they took gnotobiotic animals, that's animals that don't have a microbiome, and they gave them the microbiome from obese animals, whether it be genetic-induced obesity, diet-induced obesity, or humanized mice, that's mice getting feces from lean or obese humans. And in all cases, they found that those that got the obese had a significant increase in body fat compared to those that got the lean. And there's also evidence to suggest that obesity can alter the gut microbial composition. In these early studies, when they compared both genetically induced obesity and diet-induced obesity, they found that the lean animals had significantly less firmicute and significantly more Bacteroidetes compared to the obese animals. These types of studies have been replicated by a number of different investigators, but only when a high-fat diet was utilized, not when a low-fat diet was utilized. And I'll get to why that's important in a few slides. But while we see these effects in animals, the question is, are they relevant to humans? In this study, they compared 12 unrelated obese subjects, and they found that the obese subjects had significantly more firmicutes and significantly less Bacteroidetes than 5 lean individuals. Moreover, if the subjects were put either on a carbohydrate or a fat-restricted diet for a year and allowed to lose up to 25% of their body weight, there was a significant increase in Bacteroidetes that correlate with change in weight. So this evidence suggests that at least on an individual level, that an increase in Bacteroidetes appears to be beneficial in terms of weight. However, when meta-analysis were done comparing several large studies, including the NIH Human Microbiome Project, MetaHit in Europe, and a lot of other large studies, there was no correlation between the Bacteroidetes to firmicute ratio and body weight. And this is probably because of the large inter-individual variability among individuals. As I said before, there are a lot of studies suggesting that there's a relationship between obesity and the microbiome. However, part of the problem with a lot of these studies is that they compare animals that are fed a high-fat diet compared to those that are fed a chow diet. And perhaps they're actually utilizing the wrong comparison. So this was a wonderful study published in the last 6 months, which really tried to disentangle the effects of diet versus obesity. They had 3 different diets in this study: a chow diet, a refined low-fat diet, and a refined high-fat diet. As you can see, the chow diet and the refined low-fat diet had a similar macronutrient content, where the refined high-fat diet was very high in fat. However, they had very different fiber content. The chow diet had 15% of the diet as fiber, and the fiber came from many different sources and was a non-purified diet. In contrast, the refined diet had 5% of the diet as fiber, and it was all cellulose. And this is important because cellulose is a non-soluble fiber, so the bacteria can't actually utilize it. Here is the data. In terms of both body weight, fat mass, and glucose tolerance, those mice fed the refined high-fat diet had significantly higher fat mass, total body weight, and blood glucose concentrations, yet there were no differences in the refined low-fat diet compared to the chow diet. When we looked at the firmicutes to Bacteroidetes ratio, there were no significant differences between the 2 refined diets However, these were both different than the chow diet, both in the colon and in the feces. So what does this data mean? It means that the high-fat diet induced changes in mouse gut microbiota composition, and these effects were due to diet, not to obesity. That glucose intolerance and obesity in mice is linked to a high-fat diet, not to changes in the gut microbiota. And one of the hypotheses out there when they started showing these differences in microbial composition in animals fed the high-fat diet was that they were better able to extract energy from the diet. But this data actually shows that the cecal fermentation, the energy harvest, is decreased, not increased by the high-fat diet, as demonstrated by the decreased production of short-chain fatty acids. But the important take-home message from all of this is that the choice of diet is vitally important to studies of microbiota composition. As nutritionists, we always believe that the best diet to utilize is an AIN 93 or an AIN 76 diet because it's a purified diet. We know exactly what we're feeding. However, they utilize cellulose, which can't be utilized by the gut bacteria, and it's not going to be as an energy source for them. So there's a need to really better understand what diet you should be feeding and to make sure that all groups are fed an appropriate control diet, rather than just comparing a chow with a purified diet. So I hope I've convinced you that there's a dynamic relationship between the numbers and types of microbes and dietary components and energy consumption, that both of these can influence the microbial metabolites and specifically disease risk. But the question is can we utilize this information to predict what type of dietary changes we should be doing to influence disease susceptibility? There was a study that was published a few years that actually did this. This was a study from Israel, and they had a cohort of about 800 individuals, and they looked at their glycemic response to different foods. And what they found was that if you feed different individuals the same food, you're going to have large differences in their increase in blood sugar as a response to that food. And they wanted to try and understand, can we predict how different individuals will respond to different foods? So they utilized a combination of gut microbiome analysis, blood tests, questionnaires, anthropometrics, food dietaries, continuous glucose monitoring after giving specific foods, put this all into a computer and developed a computer algorithm and said, can we validate these results in a validation cohort, and can we do a dietary intervention study? And what they found was that there was high interpersonal variability in the post-meal glucose observed in the 800-person cohort, that utilizing the personal and microbiome features enabled accurate glucose response prediction, and in fact the prediction was accurate and superior to common practice in an independent cohort. And most importantly, when they took the 26 individuals, gave them a control diet based on how they estimated or predicted that they would respond, they actually found that the personalized dietary interventions did successfully lower post-meal glucose. While this is very interesting results, the question is always can this study be reproduced in a different population? Well, there is a company, it's called Day 2. You can go and Google them and actually provide a sample and get an app if you want to find out your microbiome and what you should be eating. But investigators at the Mayo Clinic bought the app and did an independent study in 350 people trying to see can they reproduce these results in different people and were they able to predict response and improve things. And they found that they completely reproduced the results. The studies are currently being submitted for publication. And there's also a push to do the studies are currently being conducted in a longer-term follow-up, 6 to 12 months, as well in patients with prediabetes and gestational diabetes. The other question is can you extrapolate these results to other disease conditions? So I need you to remember that when you go to eat lunch today, that you're not just feeding yourself, you'll also be feeding the microbes, and what they may want to eat might be different than what you want to eat. And that you need to take a metagenomic view of your dinner plate, rather than the traditional view. Think of all the different dietary components that can be metabolized by gut bacteria. what these metabolites are, and what their biological effects might be. So the take-home message is that microbiome research is an emerging area of science, and there are many research opportunities available. The microbiome is integral to human physiology, maintenance of health, and development of disease. That there is a two-sided relationship between diet and microbiome. And that nutritionists, dietitians, and the food industry need to stay actively informed about advances in this field. But this field still is at the preliminary stages. And most important, bacteria are your friends. And I'll take questions now. Thank you very much. So any questions? Okay.
Hi, Anita Pinjwani from Hopkins. I had a question about short-chain fatty acids and the concentration levels. So you talked about them as mostly beneficial, but sometimes they can also be seen as harmful, like propionic acid and butyric acid for the nervous system, because they have broad-range implications on gene expression and oxidative stress and inflammation. And they've been associated with like autistic disorder and schizophrenia. So could you comment on maybe if it's the difference in like concentrations, or is it maybe different methods of action that they pose?
I haven't really followed the propionic literature as much, but everything, the dose influences the poison. All sorts of specific. I was really focusing more just on the butyrate and its effects on beneficial effects in the gastrointestinal tract.
Thank you.
You're welcome. Yes.
Okay. In Western countries, and I was born in India and I'm proud American, I made an observation that we don't talk about constipation in this nation at all, which is, in my opinion, could be playing very important role in pathogenesis of many diseases, including obesity and, of course, many others.
Absolutely, and it's probably influenced by the microbial composition. And there's actually a large group at, there's a large group in a number of different societies, but are saying, are there standardized measures that we should be including when we're doing all different types of human studies to better understand changes in diet and how they're affecting microbial composition? And part of it does include stool frequency and composition, and things to be thinking about.
With respect to probiotics and dietary supplement form, how do the microbes survive the acidic environment of the stomach? Is it something to do with the formulation of the capsules?
Yes. So the question was, with probiotics, how do they survive the acidic environment in the stomach? And companies put a lot of time into looking at the best way to package them to make sure they do. In fact, one of the issues with a lot of probiotic dietary supplements is that they have to have the stated composition at the end of the half-life. So most bacterial dietary or probiotic dietary supplements actually contain significantly more live bacteria than are on the packages, just to make sure at the end of the shelf life that they actually have that number of bacteria. Barbara?
So I noted that in your presentation, you very carefully distinguished in the mouse data, certainly, between microbes detected in the feces versus microbes detected actually in the colon. So what are the latest data on the extent to which taking a probiotic, whether in food or in capsule form, can actually change the ecology and the organisms living inside your gut as opposed to what's coming out the other end?
Okay, so the question was, can probiotics in humans influence microbial composition? And the answer is, in most individuals, no. There have been a number of systematic reviews and meta-analysis that have actually looked at, are there changes in microbial composition with probiotics? And in general, no. That's not to say that for some individuals there aren't, and it probably depends on your baseline microbiota, whether how long you're taking them and the amount you're taking, what you're eating in combination. But that doesn't mean just because they're not colonizing that they may not be having beneficial effects, because they still can be exerting biological effects while they're actually there. Any other questions?
Yes. First, one little comment. At Consumer, we test the probiotics on the market. Years ago, we found that many had only half or less than they claimed. They've gotten much better. I think they realized that they weren't shipping and storing these things properly. So most recently, we're not seeing so much of a problem. My question is similar to some of the others before. So some of the most common uses of probiotics now are for taking them with antibiotics to prevent antibiotic-associated diarrhea. Do you think that's a real effect? I mean, there's lots of studies, mostly supported by industry, that show that. What do you make of that? And
I think that's one of the instances where they've shown the most efficacy. The question is still which probiotic for which individual. But there definitely is, when systematic reviews and when systematic reviews are done, they definitely in general do show a protective effect. But for a lot of the other health effects, it's not as strong. I do want to comment on your first question about the analysis of probiotics. I should mention that the U.S. Pharmacopoeia has a group of investigators right now developing protocols for analysis of probiotic dietary supplements that can be available. They're in development now to be able to actually analyze and show that they contain what they do contain.
is that breaking it down by strain?
Yes.
And is that using any DNA technology or just plating and things like that?
It's DNA technology.
Great, thanks.
Question? Back.
Hi, thank you for the talk. So there are many different techniques in identifying and quantifying the microbial composition in the community. And my question is, well, how comparable are the results obtained from different techniques, like say 16S RNA and DGG or CHIP or shotgun sequencing? Will changing a method change the conclusion by using the same sample?
Well, so the answer is that there are many different methods. And part of the problem is some of the methods will just get at which microbes are present. So like if you look at 16S, it will tell you which microbes are actually there. If you go to metagenomics, such as shotgun sequencing, you can get a better idea of biological function. So they do answer different questions, and you will get different results. Part of the problem right now, and I say this is an evolving area of science, is they're still trying to determine what are the best ways to do these studies. And one of the biggest contributor to variability among studies is actually how the DNA is isolated. So I think the important thing is within a study, everything needs to be processed the same way. Samples need to be compared to each other. Whether that then gives you the ability to compare between studies and whether you get the same results, it improves things. But there still is variability when you go from method to method. You're welcome.
In your opinion, is it more important for us to have our patients really be vigilant of what they're eating versus the intaking probiotics, or are they complementary?
I think it's more important to be vigilant in what you're eating. I think the studies with dietary fiber show that a high fiber intake is extremely beneficial. I think having lots of fruits and vegetables, whole grains, it's the recommendations that you would make for almost every specific health condition in individuals, and I still believe in terms of their effects on the microbe, it's still the best recommendations that you could be making too. Thank you very much.